What if we could diagnose an infection not in days, but in hours? Pinpoint the exact mutation driving a cancer to select the perfect drug? Or predict a genetic disease before symptoms ever appear? This isn't science fiction. This is the reality of a revolution happening right now in medicine. We're going beyond just looking at symptoms. We're reading the very code of life itself. This is the story of molecular diagnostics. Part 1. The paradigm shift. From culture plates to DNA code, defining molecular diagnostics. At its heart, molecular diagnostics is a collection of powerful techniques used to analyze biological markers in our genome, the DNA, and our transcriptome, the RNA. Instead of focusing on symptoms or the slow process of growing microbes in a lab, this field dives directly into the nucleic acid molecules that are the blueprint for health and disease. This approach allows us to answer two fundamental types of clinical questions. First, are there foreign nucleic acids present? This is the hunt for the genetic fingerprints of pathogens like bacteria, viruses, and fungi. Second, what does our own genetic code say? This involves analyzing a patient's DNA for inherited diseases or for acquired mutations, like those that drive cancer. The old paradigm and its limitations. For centuries, clinicians were limited to a generalized approach to diagnosis, lacking the tools to access the genetic information of their patients or the pathogens infecting them. This old paradigm relied on methods that, while foundational, were often slow, subjective, and imprecise. Let's consider two critical examples. First, bacterial vaginosis, BV, a common condition caused by an imbalance in vaginal flora. The traditional gold standard for testing is microscopy, using either the Amsel criteria or Nugent scoring. This process is not only time-consuming, but is also highly subjective, relying heavily on the skill and experience of the lab technician performing the assessment. A more life-threatening example is sepsis, a body's extreme response to infection that causes at least 245,000 cases and up to 48,000 deaths each year in the UK alone, more than breast, bowel, and prostate cancer combined. The conventional diagnostic method is blood culture analysis, a process that is frustratingly slow, in a condition where every hour of delay in administering the correct antibiotic dramatically increases the risk of death. These traditional methods simply lack the speed and specificity that modern medicine demands. The new paradigm, a revolution in speed, sensitivity, and specificity. We are now in the midst of a paradigm shift in diagnostics. This shift is driven by the growing recognition that molecular techniques deliver faster, more accurate, and more objective outcomes, fundamentally transforming how we detect and monitor disease. This new approach is built on three pillars. Speed. Molecular tests can identify a causative agent in hours, not days. This allows for the timely initiation of targeted treatment and effective containment efforts, which is crucial in managing infectious disease outbreaks. For a condition like sepsis, the goal is to identify the pathogen and its antimicrobial resistance profile within 1 to 3 hours, allowing clinicians to move from broad-spectrum antibiotics to a precise narrow-spectrum therapy. Sensitivity. These techniques are exceptionally sensitive, capable of detecting minute quantities of DNA or RNA. This means they can identify pathogens that are slow-growing, difficult to culture, or have already been rendered non-viable by a first course of antibiotics, dramatically improving the diagnostic yield in cases where a traditional culture would come back negative. Specificity. By targeting unique genetic sequences that act as a pathogen's barcode, molecular assays can identify the exact organism or genetic mutation with incredible specificity, reducing the ambiguity that can plague older methods. The combination of these three advantages defines the clinical utility of a test. Its direct ability to provide information that guides clinical decisions and ultimately improves patient outcomes. The philosophical shift to personalized medicine. This transition to molecular diagnostics is more than just a technological upgrade. It represents a fundamental shift in medical philosophy. We are moving away from a reactive, symptom-based model of care and toward one that is proactive, predictive, and profoundly personalized. The logic of this shift is clear. Traditional methods, with their inherent delays and imprecision, force a reactive posture. A physician treating a suspected infection must often start with broad-spectrum antibiotics, a best guess, while waiting days for a culture result to confirm the pathogen. The speed and precision of molecular tests completely change this clinical algorithm. Instead of guessing, a physician can know the exact pathogen and its resistance genes within hours. This capability shifts the central diagnostic question from "what are the symptoms?" to "what is the specific molecular cause?" Answering this molecular question is the foundational principle of personalized medicine. This approach recognizes that a one-size-fits-all model is often ineffective because of the vast variations in our individual genetic makeup and the unique molecular drivers of our diseases. Therefore, the technological leap in diagnostic speed and accuracy is the very engine driving this philosophical shift, enabling and accelerating the movement toward tailoring treatments to an individual's unique molecular profile. Part 2. The workhorse. Understanding polymerase chain reaction, PCR. The core problem, amplifying a needle in a haystack. To understand how molecular diagnostics works, we must first understand its most foundational tool, the polymerase chain reaction, or PCR. Clinical samples like blood or tissue contain a massive amount of genetic material, but the specific sequence we're looking for, from a virus for example, or a single mutated gene, might be present in vanishingly small quantities. PCR, a revolutionary technique developed by Kary Mullis in 1985, solves this problem by acting as a molecular photocopier, making billions of copies of a specific DNA segment until it is abundant enough to be easily detected. The key ingredients. The PCR cocktail. To run a PCR, a few key ingredients are mixed in a small tube. Template DNA. This is the DNA extracted from the patient's sample, which contains the target sequence to be amplified. Primers. These are short, single-stranded DNA fragments, typically 18 to 30 nucleotides long, that are chemically synthesized in a lab. They are designed to be complementary to the DNA sequences that flank the target region, acting as start and stop signals that tell the polymerase exactly which segment of the DNA to copy. The precise design of these primers is absolutely critical for the specificity of the reaction. Taq polymerase. This is the star of the show. It's a DNA polymerase, the enzyme that builds new DNA strands. Crucially, this specific polymerase is thermostable, meaning it can withstand very high temperatures. It was originally isolated from Thermus aquaticus, a bacterium that lives in hot springs. This heat stability is what made the automation of PCR possible, as the enzyme survives the repeated heating and cooling cycles and doesn't need to be replaced. Deoxynucleotide triphosphates, DNTPs. These are the individual building blocks of DNA, A, T, C, and G, that the Taq polymerase will use to construct the new DNA strands. The thermal cycling mechanism, a three-step dance, visual, animation of a DNA double helix inside a PCR tube, undergoing the three temperature steps. The entire PCR process is automated in a machine called a thermal cycler, which can change the temperature of the reaction tubes with high precision. The process consists of a series of 20 to 40 repeated cycles, with each cycle involving three distinct temperature steps. Step 1. Denaturation, 94-98 degrees Celsius. First, the reaction is heated to a temperature near boiling, typically around 95 degrees. 16. This high heat breaks the hydrogen bonds holding the two strands of the template DNA's double helix together, causing it to separate, or denature, into two single strands. Step 2. Annealing, 50-65 degrees C. Next, the temperature is rapidly lowered. This allows the primers, which are present in vast excess, to find and bind, or anneal, to their complementary sequences on the single-stranded DNA templates. The annealing temperature is critical. If it's too high, the primers won't bind efficiently, and if it's too low, they might bind non-specifically to the wrong parts of the DNA, leading to the amplification of unintended fragments. Step 3. Extension/elongation, 72 degrees C. Finally, the temperature is raised to approximately 72 degrees C, the optimal working temperature for Taq polymerase. The polymerase attaches to the primer template complex and begins synthesizing a new complementary DNA strand by adding DNTPs, extending from the primer in the 5Ds to 3As direction. Exponential amplification. These three steps, denaturation, annealing, and extension, constitute a single cycle. At the end of one cycle, the number of copies of the target DNA segment has doubled. The thermal cycler then repeats this process. Because the newly synthesized strands from one cycle serve as templates in the next, the amplification is exponential. The number of copies produced can be calculated by the formula 2 hours, wherein is the number of cycles. After 30 to 40 cycles, which can take less than an hour, this chain reaction can generate billions of identical copies, known as amplicons, from just a single starting molecule. Part 3. The evolution of PCR, adding quantification and versatility. The original PCR was a qualitative tool. It could tell you if a target sequence was present, but not how much. As clinical needs became more sophisticated, PCR technology evolved to provide quantitative answers and to work with different types of nucleic acids. Quantitative real-time PCR, qPCR. From yes-no to how much. Quantitative PCR, or qPCR, revolutionizes the technique by monitoring the amplification of DNA in real time, cycle by cycle, instead of just looking at the final product. This is accomplished by incorporating a fluorescent reporter molecule into the reaction. There are two main ways to generate this fluorescent signal. Intercalating dyes. Dyes like SYBR green are simple and cost-effective. They work by inserting themselves into any double-stranded DNA. As the PCR progresses and more double-stranded DNA amplicons are created, more dye binds, and the overall fluorescence of the sample increases proportionally. Hydrolysis. Probes. Probes, such as TaqMan probes, offer an additional layer of specificity. These are short, sequence-specific DNA strands that bind to the target region between the primers. The probe has a fluorescent reporter dye on one end and a quencher molecule on the other. When the probe is intact, the quencher suppresses the reporter's signal. However, during the extension step, the Taq polymerase's natural 5E to 3E exonuclease activity cleaves the probe as it synthesizes the new strand. This separates the reporter from the quencher, allowing it to fluoresce. The signal is only generated if the probe has bound to the correct target sequence. Visual animation showing a qPCR amplification curve with fluorescence on the y-axis and cycle number on the x-axis. A horizontal threshold line appears. The key metric in qPCR is the quantification cycle, CQ, also known as the threshold cycle, CT. This is the cycle number at which the fluorescent signal from the reaction crosses a predefined threshold, rising significantly above the background noise. The CQ value is inversely proportional to the starting amount of the target nucleic acid. A sample with a high initial concentration of the target will cross the threshold early, resulting in a low CQ value. Conversely, a sample with very few target copies will require more cycles to generate a detectable signal, resulting in a high CQ value. This relationship allows for two types of quantification. Absolute quantification. By running a series of standards with known concentrations and plotting their CQ values, a standard curve can be created. The CQ value of an unknown sample can then be compared to this curve to determine its exact starting copy number. Relative quantification. This method is often used in gene expression studies. It compares the CQ value of a target gene to the CQ value of a stable reference gene, often called a housekeeping gene, within the same sample. By comparing this ratio between a treated sample and an untreated control, one can determine the relative change in gene expression, often using the DDCT method. Reverse transcription PCR, RT-PCR. Reading the RNA message. The power of PCR is based on amplifying DNA. But what about RNA? Many important viruses, including influenza, HIV, and SARS-CoV-2, have RNA genomes. Furthermore, to study gene expression, scientists need to measure messenger RNA, mRNA, the molecule that carries genetic instructions from DNA to the cell's protein-making machinery. Taq polymerase cannot use RNA as a template. The solution is an enzyme called reverse transcriptase, which was discovered in retroviruses. This enzyme does exactly what its name implies. It performs reverse transcription, creating a single-stranded DNA copy, called complementary DNA or cDNA, from an RNA template. This newly synthesized cDNA can then serve as the perfect template for a standard PCR or qPCR reaction. When combined with qPCR, this technique is called RT-qPCR. This elegant two-step process, RNA to cDNA, then cDNA amplification, allows the entire powerful PCR toolkit to be applied to the world of RNA. Digital PCR, dPCR, the ultimate precision. Digital PCR, or dPCR, represents another leap forward, offering absolute quantification with unparalleled precision, without the need for a standard curve. Its principle is often described as divide and conquer. Here's how it works. Partitioning. A single PCR reaction mixture is partitioned into thousands, or even millions, of tiny separate microreactions, such as nanoliter-sized droplets or wells on a chip. Distribution. The target DNA molecules from the original sample are randomly distributed among these partitions. The partitioning is so extensive that many partitions will contain no target molecules, while others will contain one or a few. This random distribution follows a well-understood statistical model known as the Poisson distribution. Endpoint amplification. PCR amplification is then carried out in every partition simultaneously. After the reaction is complete, each partition is read as either positive, it fluoresces, indicating the target was present and amplified, or negative, no fluorescence, no target was present. Absolute quantification. Instead of measuring the intensity of fluorescence over time, dPCR simply counts the number of positive and negative partitions. The fraction of negative partitions is then plugged into a Poisson statistics algorithm to calculate the absolute number of target molecules that were in the original sample with extremely high precision. Because it counts molecules digitally, dPCR is much less sensitive to variations in amplification efficiency or the presence of PCR inhibitors that can affect qPCR results. This makes dPCR the ideal tool for challenging applications that demand the highest level of precision, such as detecting very rare cancer mutations in a liquid biopsy, a blood sample, accurately measuring small differences in gene copy number, or quantifying viral load with exquisite sensitivity. The causal chain of clinical need driving PCR innovation. This evolution from conventional PCR to qPCR and then to dPCR was not a random technological progression. It was a direct and logical response to the increasingly sophisticated questions being asked in the clinic. The first clinical need was qualitative, simple detection. The question was, does this patient have this pathogen? Conventional PCR answered this yes-no question brilliantly. Then, medicine advanced, and the questions became quantitative. For monitoring HIV treatment, the question was no longer just, is the virus present? But, is the viral load increasing or decreasing? For cancer research, it was, how does this new drug affect the expression of this oncogene? This demand for measuring relative change drove the development of qPCR. Finally, with the advent of concepts like the liquid biopsy, detecting tiny fragments of tumor DNA circulating in a patient's blood, a new, even more demanding challenge arose, finding a handful of mutant DNA molecules among millions of normal ones. Relative quantification was no longer sufficient. The clinical need became the absolute and precise counting of extremely rare events. This specific high-stakes demand directly spurred the refinement and clinical adoption of dPCR, whose divide and conquer method is perfectly suited to solving this ultimate needle in a haystack problem. Each major iteration of PCR technology emerged as a direct solution to a new class of clinical problems that its predecessor could not adequately solve. Part 4, the whole library, next-generation sequencing, NGS. While PCR and its variants are incredibly powerful for analyzing one or a few targets at a time, what if you need to look at hundreds or even thousands of genes at once, or the entire genome? For that, we turn to next-generation sequencing. Beyond one gene, massively parallel, sequencing NGS represents a monumental leap in scale. Also known as massively parallel sequencing, this technology doesn't just analyze one DNA fragment, it determines the sequence of millions or even billions of DNA fragments simultaneously in a single run. This incredible throughput has transformed genomics. The original Human Genome Project, which used older sequencing methods, took over a decade and cost nearly $3 billion. With NGS, an entire human genome can now be sequenced in a matter of days for $1,000 or less, making large-scale genomic research and clinical application a practical reality. The universal NGS workflow, regardless of the specific machine or chemistry, all NGS workflows follow 3 basic steps. 50. Library preparation. The process begins with extracting DNA or RNA from a sample. The DNA is then fragmented into smaller, more manageable pieces. Special DNA sequences called adapters are attached or ligated to the ends of these fragments. These adapters serve several critical functions. They allow the fragments to bind to the sequencer, and they can contain unique molecular barcodes that allow many different samples to be pulled together and sequenced in the same run, a process called multiplexing. Sequencing. The prepared collection of fragments, known as a library, is loaded onto the sequencer. Here, the specific base sequence of each of the millions of fragments is read in a massively parallel fashion. Data analysis. The sequencer outputs a tremendous amount of raw data. A sophisticated bioinformatics pipeline is then required to process this data, aligning the sequence reads to a reference genome and identifying genetic variations, to turn it into biologically meaningful information. A tour of the titans, key NGS platforms. The NGS market is dominated by a few key technologies, each with a unique approach to sequencing. Illumina, the market leader, short-read sequencing. Illumina's technology is the current gold standard for many applications, known for its extremely high accuracy and massive data output. Core technology, sequencing by synthesis, SBS, with reversible terminators. Cluster generation. First, the prepared library fragments are washed over a glass slide called a flow cell, where they attach to the surface. A process called bridge amplification then occurs, where each individual fragment is copied over and over in its own spot, creating a dense clonal cluster of millions of identical molecules. Sequencing. The sequencing process begins. A cocktail of all 4 DNA building blocks, A, T, C, G, is added. Each nucleotide is labeled with a unique fluorescent color and also has a chemical, reversible terminator that prevents more than one base from being added at a time. In each cycle, a single nucleotide is incorporated into the growing DNA strand at every cluster. A laser then excites the flow cell, and a high-resolution camera takes a picture, recording the specific color at each of the millions of clusters. The terminator and the fluorescent dye are then chemically cleaved, and the entire cycle repeats for the next base. This process happens for hundreds of cycles. Output. Illumina systems generate billions of very accurate, over 99.9% correct, but relatively short sequence reads, typically between 50 and 300 base pairs in length. PacBio, the long-read specialist, single molecule, real-time, SMRT, sequencing. PacBio's technology excels at producing very long reads with high accuracy, which is critical for assembling complex genomes and identifying large structural changes. Core technology, SMRT sequencing in zero mode. Waveguides, ZMWs. Library preparation, SMRT Bell, TM. Instead of linear fragments, PacBio creates circular DNA templates by ligating hairpin adapters to both ends of the DNA fragments, forming a structure that looks like a dumbbell. Sequencing. The library is loaded onto a chip called a SMRT cell, which contains millions of microscopic wells known as zero-mode waveguides, or ZMWs. 6.2. At the bottom of each ZMW, a single DNA polymerase enzyme is anchored, and it captures a single SMRT Bell template. 63. The 4 DNA nucleotides are added, each labeled with a different colored fluorescent dye attached to its phosphate chain. When the polymerase incorporates a nucleotide into the new DNA strand, the fluorescent dye is cleaved off and emits a brief flash of light. This flash is recorded in real time by a detector. The ZMW is so small that it confines the laser illumination to only the very bottom of the well, so only the signal from the active polymerase is detected, not the background noise from the freely diffusing nucleotides. Output: hi-fi reads. Because the template is circular, the polymerase can go around the same molecule over and over. These multiple passes, or subreads, are then computationally combined to generate one single, long, and highly accurate consensus sequence called a hi-fi read. These hi-fi reads can be tens of thousands of base pairs long, with an accuracy of over 99.9%. Oxford Nanopore, the portable pioneer, nanopore sequencing. Oxford Nanopore has revolutionized sequencing by making it portable and capable of producing extremely long reads in real time. Core technology, Electrical signal detection. Mechanism. This technology uses a membrane with thousands of tiny protein channels, or nanopores, embedded in it. An ionic current is passed through these pores. A special motor protein grabs a strand of DNA or RNA and threads it through a nanopore, one base at a time. Reading the signal, as each nucleotide passes through the narrow pore, it causes a characteristic disruption in the electrical current. This electrical signal, often called a squiggle, is unique for different bases, or short combinations of bases. These disruptions are measured in real time and decoded by sophisticated base calling algorithms, often using neural networks to determine the DNA sequence. Output. Nanopore sequencing can generate incredibly long reads, from hundreds of thousands to even millions of base pairs in length, on devices that range from the handheld Minion to large-scale production sequencers. While historically the raw read accuracy has been lower than Illumina or PacBio Hi-Fi, it is improving rapidly with new chemistries and base calling software. From raw data to clinical insight, the bioinformatics pipeline. The massive datasets produced by NGS are useless without powerful data analysis. The bioinformatics pipeline is the multi-step computational process that turns raw data into a clinical report. Primary analysis. This happens on the sequencer itself. The machine software converts the raw physical signals, light flashes for Illumina and PacBio, electrical current changes for Nanopore, into base calls, A, T, C, G, and assigns a quality score, called a Phred score, to each base. This information is packaged into a standard file format called a FASTQ file. Secondary analysis. This is where the heavy computational work begins. The short reads from the FASTQ file are first aligned or mapped to a reference human genome. Then, specialized algorithms perform variant calling, which involves systematically comparing the aligned reads from the sample to the reference genome to identify any differences, such as single nucleotide variants, SNVs, insertions, deletions, indels, or larger structural variants. Tertiary analysis, interpretation. This is the final and most critical step for clinical use. The list of identified variants is annotated with information from various databases to determine their potential clinical significance. A key resource here is ClinVar, a public archive that aggregates information on the relationships between human genetic variants and diseases. By consulting ClinVar and other resources, a clinical geneticist can classify a variant as pathogenic, disease-causing, benign, harmless, or a variant of uncertain significance, VUS, which is essential for generating an actionable clinical report. The NGS technology ecosystem. Synergy over competition. It is tempting to view these major NGS platforms as simple competitors, but it is more accurate to see them as components of a synergistic technological ecosystem. Each platform's unique strengths, Illumina's accuracy and throughput, PacBio's accurate long reads, and ONT's ultra-long reads and portability, are perfectly suited to address the weaknesses of the others. This synergy allows for a more complete and accurate view of the genome than any single technology could achieve alone. For instance, Illumina's short, highly accurate reads are the gold standard for cost-effectively finding small-scale variants like SNVs. However, these short reads are notoriously bad at resolving complex parts of the genome, such as long repetitive sequences or large structural rearrangements, which are often implicated in disease. This creates significant blind spots in our genomic analysis. This is precisely where long-read technologies like PacBio and ONT excel. Their reads, which can be thousands of bases long, can easily span these difficult regions, unambiguously mapping large structural variants and correctly assembling repetitive elements. Initially, the trade-off for these long reads was a higher error rate. This led to the development of hybrid sequencing approaches, where highly accurate Illumina short reads were used to polish or correct the errors in a long-read genome assembly, combining the best of both worlds. The technology has since evolved. PacBio's development of Hi-Fi reads, which combine long-read length with high accuracy, has reduced the dependency on short-read polishing. This dynamic creates a powerful toolkit for genomics. Researchers now select the technology, or more often, the combination of technologies, that is best suited to answer their specific biological question. The first truly complete telomere-to-telomere assembly of a human chromosome was achieved by leveraging both the highly accurate long reads from PacBio and the ultra-long reads from ONT to close the final, most challenging gaps. This perfectly illustrates that these platforms are often complementary tools, not just mutually exclusive choices. Part 5. Diagnostics on the go, Isothermal Amplification. While PCR and NGS are cornerstones of the modern diagnostic lab, they have a major limitation. They rely on complex, expensive equipment like thermal cyclers and sequencers, confining them to centralized facilities. For many situations, such as point-of-care, POC, testing in a doctor's office, screening in remote, low-resource settings, or rapid response during an outbreak, a different class of technology is needed, one that is fast, simple, and equipment-free. This is the niche filled by isothermal amplification methods. Loop-mediated isothermal amplification, LAMP is one of the most popular isothermal techniques. Mechanism: instead of cycling through different temperatures, LAMP operates at a single, constant temperature, typically around 60-65 degrees. 83. It uses a special DNA polymerase with high strand displacing activity and a complex set of 4 to 6 primers that are designed to recognize 6 to 8 distinct regions on the target sequence. This high number of recognition sites makes the reaction extremely specific. The primers initiate a chain reaction where the polymerase continuously synthesizes DNA, displacing previously made strands and forming complex loop structures. This process leads to a very rapid and exponential accumulation of DNA. Detection: the amplification is so efficient that it produces a large amount of byproduct, magnesium pyrophosphate, which makes the solution cloudy or turbid. This means a positive result can often be seen with the naked eye. For a more user-friendly readout, colorimetric dyes that change color upon amplification can be added to the reaction tube, providing a simple visual yes or no answer. This makes LAMP perfectly suited for point-of-care settings. Applications: LAMP has been widely deployed for the rapid detection of infectious diseases, including tuberculosis, HIV, and SARS-CoV-2, often working directly from patient samples like saliva with minimal preparation. Recombinase polymerase amplification, RPA is another powerful isothermal method known for its incredible speed. Mechanism: RPA is inspired by the way our cells repair DNA. It operates at a low constant temperature, often body temperature, 37 to 42 Celsius. 7. The process begins with a recombinase enzyme, which coats the primers and helps them find and invade the homologous sequence on the double-stranded DNA template. This forms a structure called a D-loop. Single-stranded binding proteins, SSBs, then bind to the displaced DNA strand to stabilize it, allowing a strand-displacing DNA polymerase to bind to the primer and begin synthesis. Speed and use: RPA is one of the fastest amplification methods available, often producing detectable results in less than 20 minutes. Its combination of speed and low operating temperature makes it an excellent candidate for rapid, portable diagnostic devices. The trade-off between simplicity and information. The development of isothermal technologies like LAMP and RPA represents a deliberate and intelligent engineering trade-off. These methods consciously sacrifice the deep quantitative precision of qPCR or the comprehensive sequence data of NGS to gain enormous advantages in speed, cost, and operational simplicity. In a centralized lab, qPCR can provide precise CQ values to quantify viral load, and NGS can deliver a full genome sequence. However, both require expensive machines, highly trained personnel, and can take hours to days to produce a result. In many frontline clinical scenarios, such as a rural clinic during a flu outbreak, the most critical question is not, what is the exact viral load to 3?